Hi, this is Nan. Today I'll be talking about developmental mechanisms conserved between invertebrates and vertebrates. So please enjoy my video.
Many developmental mechanisms are conserved between vertebrates and invertebrates. Some come into play as early as during embryonic development, maintaining stereotypical developmental patterns, while others come into play after birth, allowing organisms to adapt to their environment. One such late developmental mechanism is synaptic plasticity, which underlies one of the most important functions of life, learning and memory. Synaptic plasticity describes the mechanism of dynamic changes of wirings of neurons in nervous systems in response to neuronal activity and stimulation from the environment. According to recent research, learning-related synaptic plasticity of both invertebrates and vertebrates involves short-term and long-term changes. In both kinds of animals, the short-term strategies depend on transient increase of synaptic efficacy, while the long-term changes require activation of similar plasticity gene pathways. However, in invertebrates, those changes happen mostly in the presynaptic part, whereas in vertebrates, the changes are induced mostly postsynaptically. Now let's take a look at two specific examples of invertebrate and vertebrate plasticity pathways. The earliest and most intensively studied invertebrate in terms of learning-related synaptic plasticity is aplysia, a kind of sea slug. Researchers examined the process of non-associative learning in aplysia's gill withdrawal reflex. They discovered that pairing of tactile stimulation on the siphon and electrical stimulation on the tail resulted in an elevated response of gill withdrawal, a phenomenon called sensitization. Applying continuous electrical stimulation over the course of several days resulted in a prolonged sensitization. Let's take a look at the molecular pathways happening at the junction of the siphon sensory neuron, gill motor neuron, and the modulatory interneuron from the tail. The electrical stimulation on the tail triggers serotonin release from the interneuron, which binds to G-protein-coupled serotonin receptors on the terminal of the siphon sensory neuron and activates the G-protein cascade. The activated G-proteins then dissociates from the receptors and binds to downstream signaling molecules such as adenyl cyclase. The enzyme then produces cyclic AMPs, which in turn interact with protein kinases such as PKA and PKC. For instance, the binding of cyclic AMPs to PKA's regulatory subunits releases the catalytic subunits, allowing it to phosphorylate other proteins, including the voltage-gated potassium channels. Phosphorylation of voltage-gated potassium channels blocks potassium outflux temporarily, prolonging depolarization within the presynaptic terminal. This then allows the voltage-gated calcium channels to open for a longer period of time, elevating the calcium influx, which facilitates many presynaptic activities, such as promoting fusion of vesicles and increasing neurotransmitter release. If PKA is activated prolonged and repetitively, it can translocate into the presynaptic nucleus and trigger long-term synaptic change. The catalytic subunits of PKA can phosphorylate a transcription factor called cyclic AMP response element binding protein 1, or CRAP1. Phosphorylation of CRAP1 releases it from the repression by its inhibitory isoform, CRAP2, which then allows it to bind to its binding site, CRE. Phosphorylated CRAP1 then recruits RNA polymerase through CRAP binding protein, and a series of plasticity-related immediate early genes get expressed. One such gene encodes a ubiquitin hydrolase that facilitates degradation of the regulatory subunits of PKA, allowing persistent activity of its catalytic subunits in the presynaptic neuron. Another gene encodes a CCAAT enhancer binding protein, which is believed to play a role in forming new synaptic connections. pKa can also activate p42 MAP kinases, which can likewise phosphorylate nuclear targets. In vertebrates, intensive evidence from studies of hippocampal connections of rodents, primates, and humans suggested that similar kinase to plasticity gene pathways are presented at a postsynaptic level. In the glutamatergic synapses, the NMDA receptors play an essential role in detecting salient stimuli. Intensive signaling from the presynaptic end causes enough depolarization of the postsynaptic membrane, releasing NMDA receptors from magnesium clogging and allowing calcium to flow into the cell. Calcium influx could then activate protein kinases. For instance, it can activate pKa through the dienocyclase pathway and MAP kinases as described previously. It can also activate calcium commodulin-dependent protein kinase 2 and PKM-zeta. In the short run, these kinases facilitate insertion of new AMPA receptors into the postsynaptic membrane. In the long term, as depicted in the plegiad example, the prolonged activation of these kinases triggers the Crab pathway and new protein synthesis, among which the expression of candidate plasticity genes that are believed to facilitate the receptor trafficking. Many other proteins are also created, some of which regulate the capacity of the postsynaptic density proteins, while others promote the growth of new dendritic spines, and so on. Recent research suggested that neurotrophins and cell adhesive molecules also play a role in regulating synaptic plasticity in both invertebrates and vertebrates by providing multiple sources of calcium signaling and facilitating retrograde signaling from the postsynaptic to the presynaptic cell. This could potentially explain the function of the postsynaptic cell in invertebrates and the presynaptic cell in vertebrates during synaptic changes, and could perhaps lead to a general model across species. In summary, although synaptic change seems to happen at different pre- and post-components in invertebrates versus vertebrates, the fundamental molecular mechanisms share great similarity. The receptor kinase plasticity gene pathway allows animal to adopt a flexible responding system to the ever-changing environment.